presenter is Professor Philippe Streck. It's kind of interesting because parvovirus is not perhaps in swine medicine something that you think about as a re-emerging pathogen. Many practitioners probably don't have parvovirus top of mind. And I think Professor Streck will maybe make you realize that we need to rethink our position around parvovirus. Professor Streck is a professor in the Universidade de Caxias do Sul. I'm sure my pronunciation is terrible.
It's just perfect.
Okay, that's about all I can say. Obrigado. His background is that he's a veterinarian. He trained at the university. He also conducted a master's at the university, and then a doctor's degree at the University of Leipzig in Germany. And I believe his German is probably better than mine, which is quite embarrassing. And his focus has been around molecular epidemiology, veterinary virology, and really better understanding the viruses that affect not just swine, but with a focus on understanding the epidemiology of viruses using molecular techniques. So with that, I leave the floor to Professor Streck. Welcome and thank you for joining us today.
Thank you very much. Good afternoon. I would like to thank all the Boring Ingelheim staff for this invitation. I would like to thank also all the technical staff for the help. And I'm very honored to be here and to speak with you. And in the next 30 minutes, we will talk a little bit about this virus, porcine parvovirus. We will briefly discuss some important topics of this virus, like structure, genetic, and clinical science. Thereafter, we will see how diverse are porcine parvovirus. Then we'll take a look at the molecular epidemiology of this virus. We will see some possible evolutionary drivers of PPV. We'll take a look in the wildborns for PPV as well. And after that, we'll see some perspectives. Well, porcine parvovirus was firstly isolated in the 60s. Porcine parvovirus was found as a contaminant in cells that were used to propagate, to replicate classic swine fever. And thereafter, in the late 60s and in the 70s, PPV was obtained in several studies, always related with severe reproductive losses and also, of course, economical losses in this swine industry. To talk about the importance of PPVs is really quite easy. When I give a class of PPVs, it's quite easy to talk about PPV because PPV infect domestic swines and wildborns, and the only well-established clinical signs of PPV are the reproductive disorders that we can abbreviate with the synonym SMEDI. And for PPV, we have this inactivate vaccines in the 80s, in the late 70s, and in the 80s. We have several studies trying to establish a vaccine, and around the 80s, the vaccine was established based on this NADL2, based on these non-pathogenic NADL2 strains. At the time, for many, parvovirus was considered to be a solved problem. Another aspect of PPV that we need to know is some genetic elements of PPV. If you can take a look here in this genomic map of PPV, we can see a very small genome of about 5,000 nucleotides. In this genome, in the left part of this genome, we have these non-structural genes. They are responsible for the the replication of the virus. On the other side, on the right side of the virus, we have these structural genes, or we call it also viral proteins genes, that are responsible for the capsid. And we need to take a better look at this capsid. As you see here, at right, we have the virus capsid, this 3D structure, and this capsid is built by the viral protein. If you take a look in this capsid, you can see a triangle in there. This triangle represents the viral protein, this protein that we call it VP2. And if you repeat this protein 60 times, we have this beautiful mosaical capsid that is quite simple but also quite resistant in the environment. So this triangle represents a lot for PPV, and therefore is important to know the representation of this triangle, as you can see here in the left part of the presentation. If you see in the upper part, we have also a triangle, a 3D triangle showing the upper part of this capsid. And if you see in the inner part of this figure, we have also a triangle very down there in the figure. We have also a triangle that represents the inner part of the capsid. And in the middle, we have this figure. There is a 3D representation of the figure. We call it a cartoon technique. And in this cartoon technique, we can see some interesting parts. For example, in the inner part, a little bit down in this representation, we have this green structure, this alpha helix, and then we have this yellow structures, we have called it beta sheets. These both structures are quite important to maintain the integrity of this protein. And there we have these loops, these very large loops there in the capsid, and those loops are not so conservative because they need to interact with cells and anticorpors. Now, we know that for many, PPV was considered to be a solved problem, but in fact, it was not a solved problem. And we started to observe some genetic variability. And the first study to observe this genetic variability was performed in Brazil by Professor Hitzenheim. And Professor Hitzenheim at the time found 18 different phenotypes that clustered in two main phylogenetic groups. With different phenotypes, I mean a distinct capsid profile of this virus. Thereafter, we have some studies from Professor Uwe Truyen. Uwe also found several distinct capsid profile clustered in other distinct phylogenetic groups, and thereafter we have several researchers that also found different profiles for PPV. So, at this time, we started to make some phylogenetic analysis of PPV to understand this evolutionary history of the virus, and we observed that the virus has a very great diversity, and we also observed that we have several clusters of PPV. We also observed that these clusters have a very strong geographic relation at the time, and we also associated with that a molecular clock. And then we could observe that most of these clusters were built or were formed in the last 20 or 30 years. Therefore, PPV was passing through a very, very strong evolutionary process, a very aggressive evolutionary process. We have a lot of clusters of PPV. However, we have the dominance of certain strains, mainly these strains obtained in German, like the 27A, for example. We know that this capsid profile are quite dominant in certain parts of the world, for example, Europe and America as well. And so, for you, we have our first full question. Is it possible to observe a high mutational rate in a DNA virus like PPV? Because, just to understand the question, it is expected to have a very high mutational rate in RNA virus, like a coronavirus, for example, but we don't expect that for a DNA virus. How can I be? And please answer yes or no. And the answer was yes, we can have this high mutational rate. The first study to try to solve that question came from the team from Edward Holmes, and we have another great researchers in there like Truyan Parrish and Shackleton, and they observed that the Kenyan parvovirus originally came from the feline panleukopenia virus, and the Kenyan parvovirus passed through a very aggressive mutational rate process during the 70s and the 80s, and the virus could adapt itself into a new host from the feline to the canine. And during that time, the virus has an aggressive substitution rate quite similar to the RNA virus, what we expect in the RNA virus. Therefore, we know for porcine parvovirus that we can have, yes, this aggressive evolutionary change for the virus as well. Now, understand that PPV really mutate, that's normal, but we need to understand a little bit of these mutations. And to understand these mutations, we need to go to this very, very old school study from Peter Tyson. In that study, Peter performed a comparison between the non-virulent strain NADL2 and the virulent strain Cressy, and Peter observed that all of the differences were located only in the viral protein. Most specifically, they were located in these 6 red dots here in the figure. And if you can see these 6 red dots, you can see that these red dots are located in middle to the upper part of this protein. However, when we take a look in the new samples, in the new substitutions observed in the capsid, in the new capsid profile, for example, 27A, we can see that these new substitutions are not elsewhere in this viral protein, but are located in the surface. They are very, very, very located in these viral loops. Therefore, that leads us to think about the importance of these substitutions. Perhaps they have some importance for viral adaptation, or even they have some importance as vaccine escape substitution. Then we go to our second poll question. Is it possible that those new substitutions of the virus could reduce the neutralizing capacity of the antibodies generated by using a commercial vaccine? This very, very old commercial vaccine based on the Cressy strain, based on the NADL2 strain, that we are currently using. And the answer was yes. Let's see. The first, this problem was started to be answered by Uwe Truyen in this study. Uwe performed some non-commercial vaccine using the standard NL2 strain and other strains, and we vaccinate the sows with this vaccine. Thereafter, these sows were inseminated, and after that, the sows were inoculated with porcine parvovirus, were infected with porcine parvovirus. The result for the sows were quite okay because we could observe that we really don't have shedding of virus per feces or per saliva, and also most of the fetus were quite protected against PPV. However, when we take a look in the antibodies, in these antibodies raised from the vaccine strains in the cells, we can observe that these antibodies don't neutralize so well the novel strains, mainly the 27A strains. So, unfortunately, those antibodies have a lower efficiency to neutralize these novel strains. And at this time, the hypothesis of this vaccine escape, because of that, was quite strong. To understand a little bit more of this hypothesis and to work a little bit more with this hypothesis, we start to make some works to understand possible evolutionary drivers of PPV. And therefore, we perform two main models. We perform a model where an in silico model. In this in silico model, we perform an analysis called Bayesian approach, or population dynamic, or even skyline plot. And in this approach, the idea is to understand the number of the estimated population through the years. And we expected really to see an increase in this population number through the years. However, as you can see here, we have a decrease in this population number through years. And what was quite interesting is that this decrease was related with the introduction of vaccination in our herds. And now I need to also remember that a decrease in this population can also mean dominance of certain strains. To understand that a little bit better, we performed a second model, an in vitro model. And the idea of this in vitro model was simply to infect the virus in cells and pass this virus always forward in cells. However, we pass this virus forward using the maximum concentration of antibody that allow us to pass this virus forward, that allow us to pass an active virus forward. In other words, we pass this virus forward always in a very strong antibody pressure. And at the end of this study, we really expected to see some very nice substitutions, some very nice nucleotide mutations, amino acids mutations. However, for our surprise, in that model where we have this strong selective pressure, we really don't have a lot of substitutions. We really don't have a lot of mutations. On the other side, in the control group, without any pressure, where the virus were quite happy only with cells, we have then a lot of substitutions. We have then a lot of amino acids mutations in this model. And therefore, we started to think that maybe the vaccine was not so important for this evolution of the virus. And perhaps we have another function that we need to understand of these new amino acids. And therefore, we also perform another study. And in this study, we use the non-virulent NADL2 strain and the virulent CRESS strain as a backbone. And in this backbone, we use the technique site-direct mutagenesis or reverse genetic technique to perform a series of mutations. The idea was to create one solely mutant in each of the amino acid substitutions that we observe in strains like the 27A. So we create a series of mutants, I call it a library of mutants to observe the importance of each site. And we test these mutants obviously against antibodies, and importantly, in this time we tested these mutants against antibodies raised using some commercial vaccines, and the result was quite nice because we could see in that model that antibodies raised against the original 27A strain, they have a very, very, very good neutralization capacity. However, the antibodies generated against these vaccine strains really don't have a very strong neutralization activity. And going a little bit forward, we also tested these mutants and our system in cell. The idea was to see if these mutants possess some activity that help the cell, the virus to replicate, to replicate in cells. And the result was quite interesting because we can observe in our model that yes, all of the mutants help a lot the original backbone to replicate a lot much better in comparison to the original CRESS or original NADL2. And therefore, that leads us to think that perhaps the dominance that we see in this 27A strain, in this new capsid profile that came not from the vaccine, like a vaccine escape, but perhaps they came from a better fitness in the cells. But also, we need to understand where came this genetic diversity also. And here we may talk a little bit about wild-borns, because during that time, Professor Daniel Cadan from Romania started to perform some very nice studies with wild-borns. And Cadan always observed that this wild-born, this PPV from wild-born, they have a very great genetic diversity. It was quite difficult to clusterize these samples and also to observe sometimes some certain dominance in the samples. And when we perform a molecular clock in the samples, I mean, when we compare the evolutionary history of the samples from the European wild-borns and the domestic pigs here from Europe, for example, we can observe that the wild-borns have a much more aggressive substitution rate, I mean evolutionary rate, in comparison to the normal, to the the conventional pigs, conventional swine industry. Therefore, we hypothesize that perhaps wild-borns, non-vaccinated pigs, or even non-commercial herds may have a very, very big importance to the emergence of new amino acid substitutions. Our perspective from these studies and our perspectives for PPV, we need to remember the importance to make the surveillance and to obtain new PPV phenotypes or even new PPV capsid profiles, because we know that PPV have a very strong geographic relation. We know PPV, the distinct capsid profile may have a reduction in the neutralization capability of the antibodies raised by the vaccines, and therefore we need to use the most prevalent strains as vaccine-based strains. And finally, I think we need to understand better the role of the cellular immunity for PPV, because we know very few about this. We are just focusing in the T helper 2 cell, in cellular immunity, and we have an open field to explore in this topic. And now, at last, I would like to introduce you my parvovirus family. We have a very huge parvovirus family, and I'm glad to work with very special people like Claudio Canal, Colin Parrish, Peter Tyson, and Uwe Truyen. But also we have these new members in South Brazil. We have here assisting this seminar Jessica Maciel, Muriel Becker, and Jeza Biondo. And we are working now with Kenyan parvovirus, mostly with evolution, and we are trying to understand some very, very dramatic vaccine failures in Brazil that we have with Kenyan parvovirus. But we are also working with porcine parvovirus. We are performing some synthetic drugs and trying to understand the antiviral activities of those drugs against those viruses. Thank you very much for your time.